So I will talk about the spotlight on Epstein-Barr virus. Uh-oh. Yeah. So what is EBV? EBV is one of the most common human virus and infects 95% of adults worldwide. So the figure on my left is the EBV in the microscope, and the figure on my right is showing the structure of the virus. So in the middle of the virus is a virus DNA. It's a double-stranded DNA with a total length of 170,000 base pair that can encode more than 100 different genes. So this virus DNA is then wrapped up by different proteins. And most people get EBV in early childhood. The majority experience no symptoms or only mild symptoms such as fatigue or fever. So in some cases, EBV can cause more serious illness, such as glandular fever, the so-called the kissing disease. So how do we get infected with EBV? Primary EBV infection is typically transmitted saliva, and EBV infects our immune system, and once we're infected with EBV, it remains there for the rest of our life. So this figure summarizes a major breakthrough in EBV research. So in 1964, EBV was first discovered in Berkeley lymphoma, and in 1978, the T-cell immune control of EBV was discovered. And in the early 90s, increased EBV antibodies in MS was discovered. And 30 years later, the first clinical study of EBV-specific T-cell therapy in MS was conducted. And last year, strong evidence supporting EBV as causative in MS development. So what's the knowledge gap in this field? Despite the fact that 95% of people worldwide are infected with EBV and carry the virus for life, only a fraction of the people will develop MS. So it's not clear what determines who will develop MS following EBV infection. A better understanding of the role of EBV infection in MS development will greatly improve diagnostic, therapeutic, and preventive strategies. So in this project, we hypothesize that there are genetic variations in the EBV and the human genome that interact, leading to MS development. So how are we going to address this knowledge gap? What we learned from pandemic is that COVID have different strains. So for example, infection with Omicron will result in less severe symptoms compared with infection of Delta. And the risk of death is 60% less. Similarly, for EBV are classified into type 1, type 2. And within each type, multiple strains have been identified. So there are no differences in physical properties between EBV strains with different genetic variations. And the genetic variations in EBV are likely to result in differences in disease development. So a classic example is in a cancer called NPC. So what research find that 2 EBV genetic variations accounted for 83% of the overall risk of NPC development. So this finding have important implication for MS, is that genetic variations in the EBV may be the major drive for MS development. So last year, our team developed a pipeline that is capable of capturing nearly 100% of the EBV genome. And this provides a unique opportunity for us to really looking into the questions. And therefore, we assembled a team of diverse skills. So this including immunologists from Burgerford, computational genetics from Genome Institute of Singapore, and a statistician from the University of Queensland, and researchers from MS Research Flagship, including me, Bruce, Catherine, Nick, and Chavi, and also very lucky to have Anna and Julian, who are people with MS, join as an associate investigator in our team. So the first aim of the study is to identify the EBV strains and variations that are associated with MS development. To do this, we'll recruit 420 people with MS and 420 people without MS. So we'll collect the saliva samples, and we'll extract the DNA. We use a sequencing and genotyping technology to get the genomic information for the EBV genome as well as the human genome. So we're comparing the genetic differences between MS and the controls. And from this, we'll be able to identify whether particular EBV strains or variations are enriched in MS. For the second A, we'll evaluate the functional impacts of MS-specific EBV strains or variations on T-cell immunity. To do this, we'll select 40 people with MS. Those are with no DMT treatment, and 40 people without MS. So we'll get the blood samples, and from the blood samples, we'll isolate the immune cells with particular focus on the T cell. We'll stimulate the T cells using different EBV strain. The first one is MS-specific EBV strain that are causing the disease. The second EBV strain is the normal EBV strains. And because T cell would act differently when stimulated using different EBV strains, and we can compare the differences and know which T cell type or combination of different T cell types that contribute to MS development for the EBV infection. So the significance and impact of the project including that we will identify if there are EBV genetic variations and or EBV strains that lead to greater MS risk. We'll generate a library EBV reference genome specifically relevant to Australian, which can be used to inform EBV vaccine design. And our findings will provide invaluable insights into potential refinements of the T-cell therapy that our team has developed, which is now in a phase 2 clinical trial for MS. And here is the registration number. If you want more details, you can go to the AMZ clinical trial registry website. So the consumer involvement project, including that we work actively and collaborate with the Flagship's consumer and community reference committee. The committee are a critical part of all stages of project design, development, implementation, recruitment strategies and materials, monitoring study performance, and disseminating outcome to the MS community. So lastly, I want to wrap up my presentation by sharing a story. Back to 2008, I was selected to represent Australia to attend the Lindau Nobel Laureates meeting in Germany. So the purpose of the meeting is to provide opportunity for young researchers to meet with Nobel laureates and learn from their experience. So this figure is showing the Australian delegate, and it's for the middle photo is showing me with Robin. So Robin and Barry Marshall, they're both Australian scientists. They share the Nobel Prize for the discovery of a bacteria called HP that played an important role in the cause of stomach inflammation. They proposed the hypothesis back to the 1970s, but at that time no one believed the hypothesis. It's because the popular view at that time was that gastric disorder had a physiological basis rather than being infectious disease. But what is worse for them is that their preliminary results does not support the hypothesis. So they are not able to secure any findings to continue their work. But they strongly believe their hypothesis was true. So without any other way to do, Barry literally drank a glass of the bacteria. And within a few days, he developed inflammation in the stomach. He treated himself with the antibiotics, and he recovered from that a few days later. So by doing that, they proved the hypothesis was true. So, and their work produced one of the most radical important changes in medical perception in the last 50 years. So that is what I learned from them, is that when doing scientific research, you have to believe in yourself, because most of the time, you are in uncharted territory. And that's why I believe that my EBV work will significantly improve outcome for MS in the future. So if you want to be part of this exciting journey, please come to our training table.